University Distinguished Professor. He's also the director of the Institute for Environmental Health Sciences, and he holds the Ava Pauling Endowed Chair at the Linus Pauling Institute at Oregon State. Dr. Beckman started out his academic career at the University of Colorado as an undergraduate, and then went to Duke University to do his PhD. And many of you might not believe this, but he did his PhD studying plant sciences and biology in plants. He did postdoctoral work there, I think initially in the Division of Pulmonology in the Department of Medicine, Duke University. And from there, he went to the University of Alabama at Birmingham, UAB, where I got to know him, where he became a professor in the Department of Anesthesiology, but also in the departments of Biochemistry and Molecular Genetics, and much to my delight, in the department that I chaired, Neurobiology, where he held a secondary appointment. Along the way, he did a little stint in the military and took care of folks as a medical corps person in the military, and had a career with them as a captain in the military, and managed to integrate that into an incredibly productive scientific career. Dr. Beckman's citation lists are astronomical. ISI Google Scholar shows him something around 50,000 or more citations for his work. And for those of you that track things like that, you know how impressive those numbers are. For those of you who don't, it still sounds like a big number, doesn't it? It is. In addition, he serves on a number of editorial boards, and he's won a variety of awards, including the Medical Research Foundation Discovery Award. And as I said, he's very involved nationally in leadership positions as well. But really, the essence of Dr. Beckman in terms of his excellence and impact on the world is his own scientific research. He is truly one of the fathers and pioneers in the field of understanding reactive oxygen species and their biological effects at a whole host of levels. He has produced absolutely highly impactful, game-changing, paradigm-shifting studies and papers for understanding of the roles of nitric oxide interactions with superoxide ion, peroxynitrite formation, and the downstream signaling pathways that have such important signaling roles both in normal physiology and in pathophysiology in a wide variety of disorders, whether they be pulmonary, cardiac, renal, or neural, for example. His work has had a thread over the last few years of looking at a very devastating neurological disorder that many here are familiar with, amyotrophic lateral sclerosis, or ALS, or Lou Gehrig's disease. And he has some really exciting recent and new results that I think he's going to share with us on understanding potentially potent therapeutics by understanding the signaling pathways involved with these. And finally, I want to say that also Joe Beckman was incredibly helpful to me in my lab over a number of years in UAB, and one of the papers we had in Science at the time was a direct result of the work that we were able to get going with his tutelage and counsel. So I'm very grateful to all he's done for us as well as the whole scientific community. So Joe, welcome to Roanoke.
So I'm told I have to sing for my lunch with this. So thank you very much for having me. It's great to see you. And I feel like I'm coming to a faculty retreat from UAB. Of like being at UAB many, many years ago when it was going through a rapid expansion, and the idea of moving to a small southern industrial city was something that a lot of people wondered what you were doing, but it was a good move. It was also great to see Harry Sontheimer, and he reminded me it was 1993 when I met he and his wife and they were being recruited. And one, it's hard to believe that's a quarter of a century ago. And two, it was a really fun time. At the time, 1993 was also a really big time for me because it changed my direction, because I started to work on ALS. And it came because I was watching the national news and they announced a gene that had been discovered that caused ALS. And at the very end of it, I figured, oh yeah, it's like Huntington's. You know, what the hell is it going to do? How are we ever going to figure it out? They said it's an antioxidant enzyme, so maybe you can cure the disease with antioxidants. And I was really excited, and the paper's going to come out in Nature, but we didn't have electronic internet. So I actually went over with Dick Marquez to Mike Freelander's office to get his issue of Nature to figure out what was going on and see it really was due to mutations in copper zinc superoxide dismutase. So I'd like to do today is take you through a story where we were working on stroke and heart disease and kidney in a very general mechanism, and we've dropped it all down to studying how does superoxide dismutase cause ALS. And we've got some really exciting results, and it's really flattering to hear about all the publications, but I've also been rejected from a bunch of journals. So, get a thick skin. It's going to happen to all of you, and it doesn't matter how far you are in your career. So I'm really excited to show you that we actually may have a treatment. It works great in the mouse disease, and do we really need to worry about the human disease? The title I actually take, this also has a connection to Mike Freelander. So Mike's connected to everybody and knows everybody. So he had the head of NINDS come down once, and we met him, and I went over and talked to him, and he said, yeah, I just came from a meeting, and they said free radicals killed Lou Gehrig, and like very dismissive, and I was there, but they did. And I'm still trying to convince anybody that they really did. So let me start with the story. It's a bit faded out. There's a rainbow, and this is Corvallis, and it looks very much similar in many ways to here with nice mountains, and we even have fall colors. But the other attraction for me is that there's a lot of whitewater rivers. So this is one of the rivers we go rafting on, and Mount Hood is in the background, which I think you have to look at the other one to find it there. But you can see these snow-capped peaks in the distance. So hopefully a number of you will come out, and I've talked to some people who are working on ALS. Would love to see you out there. The other thing to mention is if you're working with zebrafish, we have one of the best facility, I think, in the world, particularly with the glioblastoma, and we're some of the first people to do that. So we can definitely help you get that model up and going. I'm teaching a course in molecular medicine, which the undergraduates are wondering what the hell did they get into. So one of the things about Oregon State is that, as I try to figure out how to even use, oh, there we go. So Linus Pauling was an undergrad at Oregon State, met his wife there, and we have a long connection with that. And among other things, he discovered sickle cell anemia. So everybody knows that this is due to a mutation that lets hemoglobin polymerize and create sickle cells. And so I've led the students along and say, yeah, and talked about complementarity. But it turns out that isn't right. That if you take out the hemoglobin from sickle cells, they still stay sickled. So it's really an oxidation phenomena. And then there's another book that's called The Demon Under the Microscope that deals with the discovery of sulfa drugs by Gerhard Thamasch, who won a Nobel Prize. It's an amazing history, and it really tells you a lot about drug discovery. And then we're also working on this book called about Vertex, a company that was founded in about 1993 is when the first book takes place. And it's about a whole new method of doing drug discovery, which actually wasn't all that new, but they had a lot of hubris in putting it together. And it deals with a lot of drugs. Basically, you can consider the protein as folded up and has nice shapes and pockets, and you design drugs to fit in the pocket. And that's what most drug design really is these days. And so what I'm going to try to tell you about is a different way of looking at a lot of pathology. This is really important. It's a key start. But really a lot of inflammation. There's a lot of oxidation that's going on. In World War I, the only thing they had to treat bacterial infections is they poured bleach on wounds. And it's too toxic to give generally. But it turns out neutrophils make tons of bleach and kill bacteria with it. And yet you go through any biochemistry book and try to find a description of chlorine chemistry or carbon-chlorine bonds, and there's nothing. And then biochemistry I'm going to talk about is even weirder. We're going to show you there's nitro groups added to proteins. You're turning your body into TNT. I actually had a panic when I first saw this and started to realize, oh my God, is this stuff explosive? And you hit the proteins with hammers and they never exploded. So I lost out on DoD grants. So I'm also going to tell you about the role of copper, which is a redox active metal and more important for redox chemistry probably than iron, and superoxide dismutase, the interactions with nitric oxide in the death of motor neurons in ALS. And I still believe that oxidative stress is a huge untapped potential for understanding many different diseases. And really we focused on ALS because it's a model organism to start to figure out what's going on. And it's relevant to everything from arthritis to heart disease to you name it. We have this new technique, and this is my friend and postdoc from many, many years ago in actually around 1993, Alvaro Estevez. And we've actually been able to prove that nitrotyrosine does something because we're using new methods that let you genetically encode oxidative damage. We've modified the tRNA synthetase to recognize nitrotyrosine, and we can put it into specific spots in proteins. And I'll show you that we've actually been able to track it down to one site in HSP90 that's enough to cause motor neurons to die. And so the story is beginning to come together in a way where we can actually prove this takes place, and we're also using mass spectrometry of entire proteins that we purify from the spinal cord in under 2 minutes to measure metal binding, and that gave us great insights to start to develop a new treatment for ALS. And because I'm going to go through a fair amount of chemistry, I want to show you what happened. So in 1993, mutations were discovered in this gene, superoxide dismutase, that I've got the paper from Mike Freelander and started to work on it. By 1994, Mark Gehrig had taken the cDNA, actually not the genomic DNA for these mutant SODs, made transgenic mice. And so the Gehrig mice have become the most widely used model of neurodegeneration. And they go out in about 100 days, they start losing weight. In 120 days, you have a mouse that looks like Stephen Hawking. And then they die at 130 days out to 150 days. So we've had this model now for almost 25 years. Thousands of labs have it. Hundreds of drugs have been tried. And there's $1 million prize if you can extend life by 25%, which was offered for many years and nobody claimed it, okay? Nobody's been able to do this reproducibly. There are some reports where it goes longer, and they're usually artifacts. So this is a common compound called copper ATSM. The image is taken from Wikipedia from the copper page. It's used as a PET imaging agent to deliver radioactive copper into tumors, and it actually can image Parkinson's lesions as well. So it's called copper ATSM. There's a group in Australia at Melbourne at the Florey that's using this to treat Parkinson's disease, and they were showing success in treating ALS as well. So we've used this in the high-expressing mice, and we've gotten the extension to 25 days, or 25%. And when I present that, the clinicians get really excited. But the result that I'm really excited about, they kind of look at you and say they don't know what to make of it, okay? So we take the SOD gene, we put it into the mouse, and you get a disease that looks remarkably like ALS. Sun and Elliott in 2007 added the copper chaperone for SOD. So this is the superoxide dismutase molecule. There's a second protein that binds to it, picks up copper from CTR1 in the membrane and puts copper into the active site. So they put in 27 copies of the human SOD gene, but there's no corresponding amount of the CCS. And it turns out when you overexpress it, the mice die 8 times faster. And I'll show you that data later. So we started to look at this in the high-expressing lines. Our mice are dying in 8 days. And I have a former grad student that was working with the copper ATSM compound. So we started treating mice. And so there are 3 mice in here that have the SOD and the CCS gene, and the other is a litter mate. And try to figure out which one. Okay, so these are 405 days. So that's 300 days past the time they would have been showing the disease. Okay, and I'll show you the data, survival data, and more about this in a minute. And then this is 550 days. They actually do progress. So you can watch the hind limbs are kind of sticking out. You can see the toes have started to spread. And these are all characteristics of the loss of some lower motor neurons. And they are now actually, we've been sacking them, but it's a remarkably effective treatment, and it's a drug that seems to be insanely safe, at least in rodents so far. So we're hoping to get into humans in the not too distant future. So I'm going to come back and talk more generally about oxygen-free radicals. And oxygen is a pretty remarkable molecule. And one reason is that it can only be reduced one electron at a time. And when you do that, you start producing intermediates or free radicals. So you generate superoxide, which was named by Linus Pauling, but it turns out it's not a superoxidizing agent. It's a weak reductant. And I'll come back to this. You make hydrogen peroxide, you make hydroxyl radical, which most people say is what really makes things toxic. But I actually think hydroxyl radical is not very toxic. And then eventually you get to the deadly water molecule, which I've seen in neurosurgery journals as written what's really the toxic species. And there's this amazing book by Nick Lane that's Oxygen, the Molecule That Made the World. And it really is really well worth reading because there are so many different implications for how the Earth forms. Oxygen, for example, the name means acid forming. And for a number of years, people thought the only way you can make an acid is by reacting oxygen with things like chlorine or sulfur to generate strong acids, which isn't quite right. Our crust is formed because oxygen atoms with metals are lighter and they float to the surface. Lots of different implications from it. This is Carl Nathan, who wrote this article in JCI. It's about specificity of a third time. And it's basically saying that reactive oxygen and nitrogen species can be very specific. So there's this thought that you form this really strong oxidizing species and it's going to react, and there's nothing further you can do, understand it. So he got kind of angry. He's an infectious disease doctor. He's applying for a joint appointment in New York to the immunology department, and they declined him because he said, well, you're working on the innate immune system, and that's not interesting. We want to know about the specificity in antibodies. But it turns out chemical reactions do have many orders of magnitude of different rates of what they react with. And so you don't have to have an enzyme that makes it specific, okay? I'm going to show you this is also the case with peroxynitrite, that what makes it an interesting oxidant is it can be very specific in what it attacks. And the chemistry, just the matter of they can be reactive. If you work out the concentration of the target, the difference in rate constants, and then the turnover of the damage, this leads to actually differences that can be tens of thousands to millions of factors different between different oxidants. And you can use your numbers and actually figure out what's biologically important. And that's what's held back the field is everybody says, oh, well, anything can happen. So, they don't worry about being quantitative. And that's led to a lot of confusion. And finally, the structure of the protein varies these factors by a factor of 1,000. That'll be hugely important because there are certain tyrosines that get these nitro groups added, far more common than other sites. So back before I even met Mike, which is really long ago, I started to work with Chung Shu, who was at the Medical University of South Carolina, and I was interested in using superoxide dismutase as a treatment for stroke. He had a very reproducible model, and we were able to show that SOD could reduce the infarct volume by about 30 to 40%. At the time, there wasn't even a good stroke model. And this took on a life of its own, working with someone actually in Richmond, and the Department of Defense got interested, and suddenly instead of working in stroke, we were working in head trauma. It actually worked in early patients. And then the FDA said, well, you have to give more and figure out what the bigger effect is. When you gave more, it turned out the brain infarct here got bigger. And if you gave it to head trauma patients, it stopped working. And so that ended that trial. So it isn't that the drug was toxic. There was something else going on. And that's been in the back of my mind of what was the toxic reaction and trying to understand why it even worked, because superoxide is not a superoxidizing agent, and we're giving SOD, and there's already a lot in vivo. So what else was happening? And then In 1988, nitric oxide was discovered to be produced by endothelial cells, and I found old chemistry textbooks that peroxynitrites formed. And this would actually, at neutral pH, break down and give you a bunch of radicals. There's a carbonate radical, which is actually far more important than hydroxyl radical in biology, but not well appreciated. And NO2 is the orange gas that you see in smog that is very damaging to your lungs. And here's the reaction that this is being formed in your body. And one of the ways we know that is it turned proteins yellow because it added nitro groups to tyrosines. And this is going to be a major theme for today. And then the chemistry side of me would try to explain to people that this is really important because it's a diffusion-limited reaction. It means every time they bump into each other, they'll react. And people would say, well, they'll never find themselves in the body. And the other is that the reaction's so fast that it's faster than superoxide, which is a big protein, can scavenge superoxide. So even if you have a lot of SOD present, this reaction occurs. The kinetics are very complex, but you can't add enough to stop it. And then the final discovery was that peroxynitrite is actually a substrate. So this is superoxide, here's superoxide. Peroxynitrite gets into the active site of SOD. And we discovered this like 2 to 3 years before I was going over to Mike's office to find this Nature paper. And this is what we thought would be the toxic principle happening in ALS before I had any clue what ALS really was. And I played soccer at UAB, and there's the other guy playing defense with me was Craig Smith, and he worked in the crystallography lab. So I guess it's actually much prettier over here. Superoxide dismutase is a really pretty protein. It's got these blue-green crystals. But after it's had peroxynitrite, it turns bright yellow. And it's still got the blue-green copper in there. And by crystallography, we found that there was this electron density increased by the 1 tyrosine on it. And that was how we came across protein SOD-catalyzed tyrosine nitration. And then the other thing we realized is you can get antibodies to phosphotyrosine, so we can make antibodies to nitrotyrosine. And that's when I finally got grants and could get tenure. And the reason is this is a human COPD, but we've stained lots of different tissues. So the brown color is the location of nitrotyrosine, which makes sense of a lot of vascular damage. There's a lot of infiltration of macrophages around it. And there are beautiful controls you could do of blocking with nitrotyrosine, or you can chemically reduce it to aminotyrosine and you lose the antibody. You can reoxidize it and the reaction comes back. You can take these tissues and digest them and isolate nitrotyrosine by mass spec and prove that it's there. So we had now an antibody in any human disease you wanted to look at, we could work on it. So this was the lung of a 2-year-old child that was healthy and died in about 4 days from an infection, and that happened a lot. At UAB would be a draw for the whole Southeast, and my friends that I was working with were intensivists, so we were studying this. Our first slides were in atherosclerosis. Over here is better. And finally, this was done, the ALS was done in Boston at the time by Neil Cowell. And you found nitration taking place in ALS. The motor neurons are stained. The white holes are nuclei. And something that I didn't pay attention to at the time is the neuropill. So there's a whole story I won't have time to get into today, but we've done a lot of work with my colleague in Uruguay, Luis Barbado, on astrocyte reactivity and macrophages and driving the progression of the disease. That would be an interesting topic, and would love to talk with Harry more about that. The other part was the specificity. So you can use these antibodies by Western blots, and what we found is we grind up the brain and add peroxynitrite to it and ask what gets nitrated. If you add more superoxide dismutase, certain proteins get nitrated. So this was actin, neurofilaments. They're structural proteins. They're very abundant and completely washed out, but maybe you can see it over here. There's a band around 90, and that's heat shock protein 90. which I never paid attention to. Alvaro recognized it, and that's a key player for what will the rest of the story from here. So I'm going to switch now to ALS and talk about, it's a crazy disease that causes paralysis, and it's due to a very selective death in the spinal cord of motor neurons. And that's shown here. Of motor neurons, you can still find them in some parts of the human brain, but there's definite pathology taking place where there's starting to be inclusions in the motor neurons. But it's only a matter of a few months that the motor neurons have completely disappeared, and the staining between these two sections is the same. There's actually a huge proliferation of astrocytes, and there's actually microglia that come in and differentiate. And the scarring that takes place in the brain, I think, is, I was amazed how little was known about it 25 years ago, and there's still not that much known. It's a great opportunity to really chase after. And then the mystery was when mutations were discovered in the superoxide dismutase, there were originally 13. I forgot to update this. It's about 180 mutations now. There's only 153 amino acids in the protein. And there are hot spots that occur for it, but it's trying to figure out how you can have mutations all over the protein and get the same phenotype. Why it's selective for motor neurons is a complete mystery, because motor neurons are the proteins expressed. It's about 0.5% of every protein in every cell in your body from before birth. And then it turns out that not everybody, it's a dominant gain of function, so you only have to have one allele to get the disease. And then what is that gain of function? And finally, depending on the populations, between 1% to 7% of populations have mutations that cause it. The disease looks pretty similar between ALS patients that have sporadic versus familial. There's far more genes discovered now, but what's the connection between wild-type SOD or sporadic and the mutant disease, or is there one, which a lot of people don't believe there is? So these are the kinds of questions I've been struggling with for 25 years, as well as the whole community. And people talk about protein aggregation being very important because it's kind of like free radicals. If proteins aggregate, it's bad for the brain, and that's Alzheimer's and Parkinson's, and we don't need to go much further. It's true that SOD does aggregate, but it is the most stable protein known. So its half-life for unfolding at 37 degrees is 237 years. And you wonder how you can measure it. Well, we've been doing the experiments for a long time. It's an extrapolation. But it was purified by the Germans and used pharmacologically by boiling cow blood in copper vats for 2 minutes. And you scrape everything else denatures and you're left with SOD. So it's one of the easier proteins, and it's also a reason I work on it, because I'm not a good biochemist, and this one is a rock-stable protein to work with. The unfolding, a huge amount's been learned about the biophysics, and what happens is that it's a dimer. When it separates, the protein weakens a bit, and then there's an order. The zinc falls out, then there's copper loss and unfolding. And the thesis that we're working on is the loss of zinc and the copper that leaves the protein toxic. Now, if you think about it, I just said I can cure the disease by giving copper, and I'm saying copper's toxic. So there's a paradox that I'm not going to be able to answer for you today, but I'll tell you that's what we're working on, and I can tell you I didn't try the copper ATSM compound for 2 years because I figured it's going to be toxic in the brain, and why bother? If there's anybody that didn't want it to work, it was me. And I'm coming back to this idea of the copper chaperone, that this domain picks up copper from the cellular membrane and it forms a physical dimer, and actually that domain comes over and drops copper in. And the final point is that the SOD has the highest affinity for copper of any protein in the brain. That's a really important, any protein in the body. There's only 12 proteins that are known in humans that actually bind copper. And the two major ones inside the cell are superoxide dismutase and then cytochrome C oxidase, the terminal oxygen acceptor in mitochondria. And what's happening is we're going to talk about an interplay of stealing copper from cytochrome C oxidase that really is driving part of this disease, okay? And we'll show you a bit more of that. So this is coming back to the study of Sun and Elliott that I mentioned before. They used a different G93A line that dies more slowly. Most of us don't bother doing it because you have to pay for keeping the mice for 4 months longer or 5 months longer. And with a low expressing line, their mice were dying in between 20 to 50 days. So that's an 8-fold extension of life, and nobody could figure out why it's happening. But they did show there was a major decrease in cytochrome C oxidase and mitochondrial pathology in these mice. So this is a mass spec lab at Oregon State. This is Jared Williams, who's a grad student who's done most of this work. Nathan Lopez, who's a technician in my lab. Blaine did his PhD with me and went to Australia because he met our star volleyball player who was on the Australian volleyball team, and it was the only reason we got him at OSU. And then Erin was from New Zealand, and she was a grad student in Melbourne also. So they came over, and this is about 3 years ago. We were, I'll show you the experiments they were doing later. And we had these mice that were dying very rapidly from the CCS expression, and they were talking about this copper compound. So we made it and tried it in the mice and got absolutely amazing results. So here's the survival of G93A mice, the high expressing line. And if we cross the CCS to it, the mice are runts, and they're dead within 8 to 12 days. And so it was those mice that we treated with the compound. And this is the compound. It looks really weird, sulfurs and lots of nitrogens. So I describe it as you make it by starting with a chemical that killed the people in Bhopal, India, mix it with rocket fuel, and then you add popcorn smell, and then you add copper, and it comes out as a pure compound. And you can get it done in about 2 hours. So it's a crazy compound. They call it brick dust because it appears to be incredibly hydrophobic, and it's orange, and it's a powder that floats around. And it's soluble in dimethyl sulfoxide, and the question is, how do you treat a 5-day-old animal with it? Well, it's on DMSO, and DMSO crosses the skin, so this is not a drop of blood on the skin of a mouse. That's the compound. Drives our vivarium people crazy when they first started to see this red compound on the cage, trying to figure out which animal bought which and left blood. If you look after 5 minutes, it's absorbed through the skin. 10 minutes, the skin is no longer reddish, and then it's in the fat, and it's already distributed into the brain by then. So it becomes easy. I can pipette the drug on mice, and we've been doing that for 3 years. And then to evaluate it, you can see it. So blinding of experiments is important. This is Nathan Lopez, and you can see this orange tinge on the coat. And Nathan realized these laser goggles that are bright red can be used. So I'm not going to tell the randomization that we used in the trials. I'll show you in a second. But they were carefully randomized, checked with a statistician, and we did the evaluations for a while that were by this blinded method. You can see the glasses. I had one paper rejected, or this paper was rejected from Science because we didn't do the whole study as blinded. And the reason was, after 200 days, why go through this rigmarole? It was clear the drug had worked far better than, because these were the results. I used this one. So the top is a Kaplan-Meier survival curve. So these mice die in 8 to 10 days. If you give the drug on the black line continuously, they've made it out as far as 680 days, and there's 2 mice still alive. There's 17 mice in that group. When we started to publish it, all the mice were alive, and that was over a year of survival. The weight is also impressive. That the black line, you can see going out, that the mice actually became obese. Weight loss in ALS is actually the worst prognosis you have. So our mice getting fat was a worry, but it's not a bad thing. Then you can give the drug for 21 days till the mice are weaned, and take it away. And what happens is the mice still grow fine for 2 to 3 months. Then they start getting hind limb symptoms, lose weight, and they all die at 130 days. It looks very much like the other mouse, the standard line, but it's not exactly the same. We can then take the mice after they're sick and start to deliver the drug again, and then those mice survived out to about a year. And half of them developed ALS symptoms, others died of other causes. So we're able to stop the disease, restart it, and keep them alive for many, many months past where anybody has done it before.
Sure.
Yeah.
Is there, I don't know how often you tried to stop-start, but is there some critical threshold where the re-onset of symptomology is a different kinetic, like there's some cumulative beneficial effect that appears, or it's just like starting fresh every time you go all the way back?
So these animals are symptomatic. It takes a long time. You can see that it took a while for weight to start to gain. They were definitely symptomatic, but they did improve symptoms quite a bit. We've done it in different ways, and this is the high-expressing line. We use the low-expressing line now to follow the longer course. So you can definitely rescue them in a number of different ways. But I haven't had funding for 3 years either, so I've been paying for these mouse charges myself to do these studies. So there's a lot we haven't done with it too yet. And to be honest, I'm looking for other drugs that would work better or other variants, and that's where we put a lot of our effort. This is just repeating. The experiments were randomized, and we ran same parents and showed that we had this 25% extension of life if we treated mice very early with, and they didn't have the CCS gene. So it's not, there's TV movies of, look, the mouse is still alive at a year, and it's pretty famous. It was a glutamate transporter that was supposedly overexpressed. And the embarrassing part is the mouse had lost the SOD gene. It was not a transgenic, so that's why it was still alive. We have done PCR on all these, and the transgene's there, but we're also measuring the SOD protein, and I'll show you that the amount of protein doubled. So it's not like we got rid of the protein, we're actually stabilizing it. And that's why I'm going to show you a couple of things we've done from here. We've done histopathology, and when the animals become symptomatic, there's definite motor neuron loss shown down here. And most importantly, we get tyrosine nitration. That's my only marker I trust. There's astrocytosis, and then this all reappears after you take away the drug or if the mice became symptomatic. The other experiment that we've had this data, but I hadn't looked at it till a few weeks ago and suddenly realized, wait a minute, this is really cool. So we're measuring cytochrome C oxidase. In the animals that we withdrew the drug, they have a quarter of their cytochrome C oxidase left in brain and spinal cord. There were some preliminary experiments we did with low doses where the animals got sick at around a year, just under a year. And their activities were really scattered all over the map, but they were animals that became symptomatic. And we were originally trying to use a very low dose of the compound because it's copper containing. We didn't know what the toxicity would be if you keep giving it. And then the other thing that we've been working on, you're going to have to use the one over there, is a method of using mass spectrometry where you can get the entire protein into the mass spectrometer and leave the metals bound to it, all the post-translational modifications. And so the method is you take a biopsy punch that you use to take nuclei out to measure neurotransmitters in the brain. And we drop that punch into a little bit of buffer and mush it, and then we pull it up onto a pipette tip that contains C4 HPLC packing. It's called a zip tip, and it's used in mass spectrometry to clean up peptides. Well, it turns out SOD binds to it. You rinse it with water, you get rid of the lipids and the salts, and then my technology innovation is whittling out a piece of tubing and putting it into the mass spec, and you flow the sample straight into it. So the bottom is a mass spectrum. It's deconvoluted, but basically we're weighing the proteins that come out of the brain. And the SOD, you can see 3 spikes, and we've added bovine SOD as an internal standard. So think of it as running an SDS gel, but taking 2 minutes to prepare the sample, running the gel in a minute, having a resolution to better than 1 Dalton, and keeping the post-translational modifications bound to it, okay? So I think it's pretty cool. I get dinged at NIH because we're not being innovative. And the other part is we've done Western blots to show that all the SOD is still in the soluble fraction, that it's not, they talk about aggregation. Well, it turns out it's less than 1%, and that's only at the very end stage of the disease. So we're not missing much of the protein. So the result here is that we're measuring the SOD, and a huge amount of the SOD is missing one metal, and it's missing zinc. And it turns out what's different is it's in the spinal cord that you're just not able to get enough copper into it to populate all the SOD that's being made. The brain is second, and then other organs, copper is not quite so limiting. So that's one distinction of what's different about the spinal cord than the rest of the tissues. We're actually able to do this method using laser capture, and we can measure SOD from 200 motor neurons. There's not much difference between including the neuropill. It's not specific to motor neurons. But that zinc-containing copper-deficient SOD is not toxic because wild-type mice that don't get the disease, they have more of the protein that's missing copper. So what I'm going to show you is we're treating the disease, but the biggest obvious peak is not the toxic species. So I'm going to move on forward because we've talked more about copper ATSM. There's also biochemically a really sound reason for this, that the SOD protein, if it doesn't have metals, is one of the most unstable proteins known. And I told you it's the most stable protein, but that's with its metals seated. And it turns out adding zinc, it accelerates the folding of the protein 100-fold, and it provides 13 kilocalories of stabilization. So it really puts it off the chart, and this is work of Matthews, who's up at UMass. So this is showing the mass spec that we're using now. There's little spikes, that's weighing SOD, and each spike is difference of 1 neutron because we have carbon-13 and carbon-12. So that changes the isotopic distribution. It's not one peak, but a series of peaks. And what happens is the peak that's over here, that's missing the copper atom. And when we have CCS present and add copper ATSM, that drives it all to the mature copper zinc form, which is really stable. And you need the CCS to do this reaction. If you just give copper ATSM, you get some conversion, but not very complete. So this is just showing what our typical peaks look like. And if we give copper ATSM, the thing that is really remarkable is how small this peak is. I've never seen this happen or get that small. And it happens in about 3 days. So it's a very effective and rapid transformation. And then the quantitation will be hard to see, but the amount of SOD protein here is phenomenal. That we have almost 200 micromolar when the animals are dying in the G93As. Now you only have 30 micromolar oxygen in the brain, so this antioxidant enzyme is 7 times more when it's overexpressed. Endogenous SOD in humans and mice is about 4 micromolar. In our animals that have been treated with copper ATSM, it's about 320 micromolar. We've doubled the amount of protein, but it's all in its mature copper zinc form, its active form, or almost all there. So now I have the paradox of I'm curing the disease by giving copper, and yet I've built most of my career saying copper in SOD is really toxic. And there is a way to make it work, but I'm not going to get completely there, but I'll show you where we're going. But this is just showing that what we've been doing since the 1993 is purifying SOD. So the green is copper zinc SOD, one's wild type, one's mutant. If you take away the zinc atom, it turns to blue. And Blaine Roberts and John Tainer did the crystal structure, and if you lose zinc, you can see this incredible disorder where there's a large part of the protein you can't see, but you can see the copper becomes visible, okay? So there's a loop that's coming up and it's floating around, it's really flexible on this protein when you lose zinc. And that's going to be a really key player for this. And then this is Uruguay. So Alvaro Estevez, I mentioned, he's now down at University of Central Florida. Patricia Casina and Luis Barbado, who are working on the astrocyte part of the story, and others. And they're all in Uruguay. And they've been culturing motor neurons in a dish. So these are motor neurons on an astrocyte feeder layer. And it's been a great system to study that we actually get absolutely trashed for. And now everybody's coming back to do iPSC cells and saying, oh, it's ALS in a dish. It's like we have all this stuff, many, many papers characterizing it that are just being ignored. So you need expensive trophic factors to keep motor neurons alive. And what happens, this is what Alvaro did in 1994, if you take away the trophic factors, the motor neurons die, not surprisingly. But if you inhibit NO production, or you give scavengers of superoxide, you block this death, okay? So the crazy thing is they make peroxynitrite, and that induces apoptosis. Why that happens, I would never have believed this would be the mechanism, but we've been doing it for 20 years. And we did get a paper in Science. It was the last paper of the last millennium, so our work is now a millennium old. But basically, you can show that even wild-type protein, if SOD, if it's missing zinc, will kill motor neurons, and you see an induction of nitrotyrosine, okay? So we've got lots of ways of working on the model system. We know that the motor neurons become nitrated before they die, and then have been working through the different techniques to work out what's going on, and actually, how do you ever prove that nitration is important? So Alvaro also started doing some crazy experiments where there's a list of proteins. If you do mass spec, you can see what has nitrotyrosine on it. And tubulin, actin, I showed that a long time ago, an elongation factor, HSP90, HSP70. So he took each of these proteins as a pure protein, treated it with peroxynitrite, and then started to add it back to motor neurons. And the amazing result is if you added peroxynitrite to HSP90, you got as much cell death as you would get by the endogenous mechanisms. And nitration of the other proteins had no effect. Now, the protein had to be delivered with chariot to get inside the cell. If it's outside the cell, it doesn't do anything. It's a critical protein for 300 clients. But the center domain here binds unfolded proteins. And it turns out when you have zinc-deficient SOD, that loop that floats around lets it bind with high affinity to HSP90. And the copper-zinc form doesn't bind at all. And it turns out the nitration sites that are important are two sites near the ATP binding site. If you modify either of them, it makes HSP, gives it a gain of function that induces the death of motor neurons. And to make a large story, or to try to explain what goes on, is we know that if you take away trophic factors or have alter signaling mechanisms, you can turn on NO production. That's not toxic. You turn on a pathway that involves ceramide to produce superoxide. The two together induce tyrosine nitration, and that leads to an apoptotic cell death. HSP90 appears to be a key player for the nitration, and if you just put in nitrated HSP90, that will induce the cell death cascade. There's a lot of things. How do you know it's tyrosine nitration? This is where we created a center to genetically encode HSP90. So we make the protein in E. coli. We put in a tRNA synthetase that will recognize nitrotyrosine and not tyrosine. And you make a suppressor, put in a stop codon in the gene for HSP90 that this tRNA recognizes. So you can put a nitro group, nitrotyrosine, at each spot in the protein, and you can make purified protein separately, and we added each of those back, and it was tyrosine 56 that's involved that will induce the death cascade. We tried to publish it in Cell and got into this 2-year battle where they said, oh, you have to find the target. And we had a lab meeting. There was a graduate student working on P2X7, which is an extracellular ATP receptor and really important in a lot of glial and microglial processes. Turns out it's also expressed on motor neurons. And there's one paper that reports HSP90 forms a complex that keeps P2X7 in an inactive state. So we went through and did a bunch of experiments. We showed that nitrated HSP90 activates P2X7. That lets in calcium, and that was really important for the death cascade. So all of this was published in PNAS in 2013. And then another key player in it is also FAS ligand. We also have antibodies that recognize different parts of it, but the one that I really like is as animals become symptomatic, this is the antibody for that recognizes just that tyrosine on HSP90 becoming more and more positive, and this is true in mice, rats, and humans. There's far more stories, like nitration of NGF is also involved in this, and it's being nitrated in astrocytes, and that's inducing death. So the summary is that I argue our CCS mouse is actually a humanized model of ALS because every human that has this disease has CCS in 2 gene copies and 1 mutant. So we're just reestablishing the balance. The reviewers argue that it's not a model of ALS at all, and so forget it. I haven't talked about this, but actually there's a lot of good evidence that what's happening with the SOD mutations is they frustrate metal binding. What they do to the overall protein is they just disrupt it enough. Sometimes they stabilize the protein, but it's very clear it interferes with the proper insertion of metals. And so that's what the gain of function is. It's related to metal binding in some way. Then there's a huge amount of the protein because it's overexpressed in the spinal cord that doesn't have copper. That makes sense because copper import into the spinal cord is extremely slow. The turnover of copper in a mouse brain is over a year. It just doesn't get in and it doesn't get released very often. And we need CCS to load copper into SOD. And if it's there in these mutant mice, it's sucking copper away from cytochrome C oxidase. That's creating the early death. And then if we give the copper ATSM, that's able to get copper into the brain and reload SOD and also replete and activate the cytochrome C oxidase. But copper ATSM is doing more than just delivering copper. It works in many models of Parkinson's disease, which aren't due to a copper deficiency, and we're trying to understand some of that pathology. I want to take, running long, but I just want to show you, we've been playing with mass spec further. And we've invented a device that gets put in the mass spec and involves a light bulb filament and 2 magnets. And it holds electrons right in the path of ions. Because the problem I have is, can I actually localize where a metal is bound in a protein? The same's true if you want to measure phosphorylation. It's really tough to do by mass spec because the fragmentation methods knock off phosphate groups. And so what we've come up with is a method where with this device, this is the hot filament, and these are the magnetic field lines from the magnets. And what happens is if you put a voltage on, you can start to suck the electrons off the filament, but then they get parked out here and stuck, so they have very low energy. And that's perfect for binding to proteins at the peptide bond. So when proteins fall through, they, by the time they traverse the cell that's only a centimeter or 2 long, they become fragmented at one spot. And it's like creating a DNA sequencing gel that you can sequence proteins. And you can do it now in like 30 seconds. In principle, we can do it in 1/10 of a second. So this shows the copper zinc protein, and there's copper bound on one end of the protein and zinc's bound on the other. So in a case, if we can put an electron right here, we can actually separate the two sides. And then the electron binds to the peptide bond, it creates a break and separates into two pieces. And it's a quantum effect so that there's not energy being put in. Collision-induced dissociation involves hitting it with a baseball bat, and that knocks a lot of things out. Here it turns out the metals actually stay bound to the protein, and this was the shocking result. So we've isolated the protein, and you can actually purify it in one part of a mass spec and pass it on to the next part. And then when we turn on the electrons, you get what looks like this incredible amount of noise. But if you amplify it, probably you have to look over here, it turns out that each of those is a piece that's exactly predicted from its sequence. And the multiple spikes here, these are isotopes having different amounts of carbon-13. So these are two proteins, one that's missing copper, one that's missing zinc. And you can see in places where we can find copper bound at one site, but not, and zinc on the other protein. So the reason why we spent 10 years building these machines is I can actually start to take SOD now and figure out, does it have copper in it, does it have zinc? Because you can't do it separately, and there's been no other method where you could begin to do this. So I don't know what the final answer will be, but we're having fun with it. So anyway, there are a lot of people that contributed to this, and I'll be happy to answer questions. Yes?